Hi everybody. In this video we're going to be talking about the topic of biotechnology. The first aspect of biotechnology I'd like to discuss is genetic engineering. This is the manipulation of genes and gene products. The individuals pictured here and here are Herb Boyer and Stanley Cohen. They performed a landmark experiment in which they moved a gene from a frog into bacterial cells. This created the first transgenic organism. A transgene event means that we're moving a gene across species barriers. So again, you're taking a gene from a frog, one species, you're moving it into bacterial cells. This is a different species. In a landmark decision, the U.S. Patent Office declared that Boyer and Cohen could patent this new transgenic bacteria that had the frog gene in it. So what are the important tools that scientists use in biotechnology experiments? So let's talk about recombinant DNA tools. The first really important tool are restriction enzymes. These are proteins that cut DNA at very specific sites. Typically, restriction enzymes recognize what are called palindromic sequences in the DNA. The example in this image looks at EcoR1 or EcoRI. This recognizes a palindrome sequence of GAATTC, and it cuts the DNA in a way which produces not a cut straight through the molecule, but produces what's called a sticky end. So different pieces of DNA cut by EcoR1 could pair up in new ways, and they're going to stick together because of complementary base pairing, A's with T's, G's with C's. Cloning vectors are carriers of genetic material from test tube to host organism. A very common cloning vector is plasmid DNA. Host organisms are where you're going to move the gene that you're interested in. Typically, genes are moved into bacteria because bacteria are very cheap, they're inexpensive to grow in the laboratory, and there's the positive thing that they reproduce very, very quickly, so you can grow huge numbers of bacteria in a very short period of time. Other important techniques include PCR, polymerase chain reaction, which allows you to take a small sample of DNA and produce lots and lots of copies, millions of copies of that DNA. Finally, DNA sequencing. These techniques allow scientists to determine the exact nucleotide base sequences in a DNA molecule. So what is the order of all the A's and T's and G's and C's present in a sample of DNA? This image shows a number of examples of different restriction enzymes, and the point is that there are so many of these that if we have a piece of DNA, and my pen is going to be my stand-in for a piece of DNA, let's say I have a gene that's from right here to right here that I'm interested in. It's a very good likelihood that I can find a restriction enzyme that's going to cut just upstream of my gene and just downstream of my gene, so I can cut out just that portion of the DNA molecule that I'm interested in, and then hopefully later I can place that into something like a plasmid and then move it into bacterial cells in order to do some cool experiments. Let's take a closer look at how plasmids would be used to clone genes. So what you need to do first is you need to isolate a plasmid from bacterial cells, and you need to isolate DNA, which contains YFG, your favorite gene, the gene of interest. You're then going to cut them both with restriction enzymes, which allow you to recombine the DNA in new ways. So then you're going to produce a new plasmid, an artificial man-made plasmid, which has your favorite gene inserted into it. That plasmid could then be placed into a bacterial cell using a technique called transformation, which uses heat shock and some salt solutions to move plasmid DNA from outside the cell to the inside of the cell. Now you've got cells, hopefully, that have this plasmid contained inside of them. You would then try to identify desired clones. Normally, when artificial plasmids are created, you're not just putting your favorite gene into that plasmid. You're going to put other genes which would give cells unique traits that other cells don't have. For example, the plasmid might be including a gene that would give antibiotic resistance to a bacteria. So after you've produced your cells that hopefully have the plasmid added into them, you would try to grow them on a petri dish that contains the antibiotic. If the cells grow, you know that they have the plasmid you tried to put into them because the plasmid gives them the ability to be resistant, to grow in the presence of that antibiotic. If they grow with that antibiotic there, they've got the plasmid, which also means now they've got your favorite gene included in them as well. There's a classic experiment that many students do. It comes from Biorad, the company Bio-Rad. It's called P-Glow. So it moves a plasmid into bacterial cells, which gives them resistance to the antibiotic ampicillin. It also gives the cells the ability to glow when they're exposed to UV light. The Human Genome Project was another really landmark development in the field of biotechnology. What happened was this. First, scientists started to develop linkage maps of the human genome. This means that they were identifying which genes were located on chromosome number 1, so they would all be linked together on chromosome number 1, which genes were located on chromosome number 2. This allowed for some mapping, physical mapping of the genome, which genes were on which chromosomes. Eventually, the desire turned to sequencing, getting the letters out, the A's and T's and G's and C's. What's their order? There's 3 billion base pairs in the human genome. So this was a huge project to try to figure out the order of all of those base pairs. The genome sequence could then be analyzed to locate genes that hadn't been discovered yet, to compare the human genome with genomes from other species. Here we see a picture of the lead individuals from both projects who are working on sequencing the human genome. This is Craig Venter from Celera. This is Francis Collins, who was head of the government project. Here they're pictured at the press conference with President Clinton when the two projects basically took co-credit for sequencing the human genome. Now, what's coming after the Human Genome Project? Most scientists are looking at the human proteome. This would be to develop a full understanding of the sequence and shapes of all the different proteins that function in our bodies, which give us enzymes, which give us molecules that move things in and out of our cells. So much of what we are is determined by the proteins that we have. So if we can better understand the proteome, we can better understand the functioning of the human body, and then along with that, we may be able to in the future cure diseases that have no cures at this time. Let's take a closer look at the technique called polymerase chain reaction, or PCR for short. So here's how it works. Imagine you're a forensic scientist and you're analyzing evidence from a crime scene, and you find a biological specimen. Maybe it's a blood spatter. And so that blood can be taken, and if you're lucky, you'll identify DNA from that blood. Now, there might not be a lot of DNA from that blood. So to really to allow you to look at that DNA and to try to use it to try to identify maybe a suspect or someone who perpetrated a crime, you want to make lots and lots of copies of that DNA so you can better analyze it. So here's how PCR works. You're going to take that target DNA and you put it into a thermal cycler. That is going to use heat to make the DNA double-stranded molecule pull apart from each other. It includes a heat-stable DNA polymerase enzyme, so it doesn't denature with these high temperatures. That DNA polymerase is going to make copies of the strands that were pulled apart. There's all the nucleotides that you need to make more DNA are present in the thermal cycler. Now, this process can be repeated multiple times. So you heat the DNA, pull it apart, make copies. So you go from one DNA molecule to now you've got 2 DNA molecules. Go through another round of PCR, you go from 2 to 4 molecules, and then from 4 to 8 after another round, and then from 8 to 16, and then to 32, and then to 64, and 128, and before you know it, you have a huge number of DNA molecules. You can produce over 1 million copies of interesting DNA in a very short period of time using a PCR thermal cycler. This is the end of part 1 of this lecture presentation. Please use the link below me right down here to continue on to part 2. Thanks for watching, everybody.